again, and thank you for joining us here in this stream. This is the Bioinformatics workshop organized together by HiDucator and NUIG. This week, we have worked through a substantial amount of material, learning a lot about the most fundamental bioinformatics tools. Thank you for being active on Moodle. As you have noticed, practicing to use the demonstrated bioinformatics tools is an essential component of this course. Regarding the homework assignments, please pay attention to the deadlines. Some of those are strict. Today, we turn our attention to a large but very important segment of bioinformatics analysis, namely working with three-dimensional molecular structures. Dr. Tolvanen will introduce you the basics of protein models and the central database dedicated for this area, PDB, the Protein Data Bank. Welcome.
Good morning again. We are soon approaching halfway through our workshop, and it's time to change gears. We'll move from sequences into structures, namely three-dimensional structures of proteins, and we'll see the database called PDB. Before we go there, I'm going to go through a couple of things of studying of protein structures. This might be repeating things you already know, but just as a prelude for the PDB, so we'll look at these things. Then we'll look a little bit inside of PDB files to see what it is like as text. That will help you understand how it's rendered into graphics, and also will give you an idea how to select things when you see how things are called within the file. These same things will be usable in molecular graphics. And on Monday we'll be moving to actually making some graphics, but as of now we'll just look at the data sources. We'll look at some search strategies, how to find data in PDB, and then finally we'll start to prepare for making images by showing what kinds of images are common to use for proteins. So how is it that we can know anything of protein structures? They are really below even nanoscale of things. They would be measuring something like a couple of nanometers across, and so there's no way that we can see them directly. So in electron microscopy they would be mostly seen as blobs, and with some very special methods like atomic force microscopy we might get some idea of the shape of a surface of a protein. So what we know about protein structures comes from very indirect complex physical methods. The most common and best in terms of resolution is X-ray crystallography. There's also neutron diffraction, but there are not that many structures that we can see. There's NMR, nuclear magnetic resonance, that is quite commonly used for proteins, and then something that used to be a low resolution method but is now approaching atomic resolution. It is cryo-electron microscopy, which means image reconstruction from single particles, or rather electron micrographs of single particles, viewed from different angles or maybe viewed from different particles. There are now efficient mathematics to do the reconstruction of 3D structure from that. And this cryo-EM is really a method of future, because it's filling in a gap in the abilities of X-ray diffraction and NMR. Both of them are limited by molecular size, so that large proteins and protein complexes are much more difficult. And in cryo-EM the problem is the opposite. It doesn't do well with small proteins, single proteins, but it's very efficient for very large protein particles. Now, this is like a 1-minute crash course in protein crystallography. You have to crystallize a protein, which might take somebody in the lab several months before they start getting crystals. Then you radiate the crystal with a narrow beam of X-rays and you get a diffraction pattern. You do that from multiple directions. You tilt your crystal to get more of those dots, and then you have tens of thousands, maybe even hundreds of thousands of these diffraction dots. Their intensities are measured, and with some fancy mathematics and a problem in figuring out what are the phases of the X-rays, diffracted X-rays, you finally would have an electron density map coming from inverse Fourier transform operations. And once you are there, the electron density can be fitted with the protein sequence, and you get an atomic model of what you wanted to study in the first place, the protein you crystallized. And then you might be iterating between creating the phases with the help of an improved model, get an improved electron density and remodel and so on. Now in cryo-EM, the resolution can be pretty easily visualized from the models that people have generated of the same protein complexes over the years. Way back decades ago, you could only get a resolution of 17 angstroms, which means that you can't really figure out anything of atoms, not even secondary structures. And with improved resolution, you can see more and more detail. 3.9 is still like terrible for compared to X-ray crystallography, but nowadays they go even to 2 angstrom and below, which means atomic resolution and models which are really dynamic. Sorry, they are taken from real live particles instead of being crystallized, so you could consider them to be even more natural state of the protein than X-ray crystallography. But anyway, this is getting better and better, and this is a really fast method. You only need those particles in solution. You don't need to crystallize or even purify at a very high degree. So this happens quickly, and there are more and more of these devices and software setups, so that they have been increasing in the past few years. And this might mean a real revolution in structural biology. Now, where can we find protein models? I've advertised PDB, the protein bank, in many occasions, and this is where we should go. It is a database, or rather a database consortium. We have a link to worldwide PDB project in Moodle, and you'll find the participants there. So it's a repository for three-dimensional structures of biological macromolecules, meaning basically proteins and nucleic acids and their complexes. We don't have large collections of small molecules there only if we happen to have a drug or a ligand, which is a small molecular compound, bound to a protein. They would be around as byproducts. If you want small molecules, there's Cambridge Structural Database, which is commercial, and PubChem, which provides three-dimensional models of smaller compounds. Now, what do we have in PDB? This is a nice table in that it gives you both information of the experimental methods and the targets, what kinds of molecules. So if we look at the total numbers of molecular types, you can see that protein only is the most common kind. Then we have glycoproteins, so proteins with oligosaccharides, nucleic acid and protein complexes, and only a few % of the content is something which doesn't have any protein in it. As to the methods, so X-ray crystallography 154,000 out of 174,000. It's it's almost 90%, not quite. Not far enough. NMR has less than 10%, and these EM-based models are about 4% of the total content, and everything else is just tiny fractions of all the data. Well, we've learned that X-ray crystallography is really popular and we have a lot of models there. So we need to know some technicalities to understand how they describe the models in PDB so that we'll be able to pick up the best ones out of multiple ones which we may find. So two very easy measures for the quality of the structures, or rather models we have in PDB, can be compressed into just two numbers. There's resolution, in which you have to know that smaller is better, and it's an indication of how much data was used to create the model. And its unit is angstroms, which is a measure of length 10 to the minus 10th meters, or 0.1 nanometers. So a resolution of 2 angstroms is pretty okay and kind of average or median resolution in all of PDB, and 2.5 is starting to be a bit suspicious. You can't really know if your amino acid side chains are where they are supposed to be in the model. There's a lot of uncertainty at such a poor resolution. Here's a link to more information about resolution in case you're interested. And then there's another thing called R factor, which is a measure of how well the model fits the actual data, the diffraction data, or structure factors, as they call these individual intensities of diffraction points, including the coordinates in the diffraction image. And this R factor should be approximately 10% in the numerical value of the resolution in angstroms. So for a 2 angstrom model, the R factor is expected to be about 0.2. And I'm talking about models in PDB because even if they are based on experiments and they have a basis in reality, they are still the best representation they can build for the electron density, which again is an interpretation of the original diffraction data. There are many steps and uncertainties so that we don't know for sure if all parts of the model are actually true, but instead it is just the best way of representing the diffraction data as an atomic model that probably created the diffraction. Now, when we go to PDB files and open them for visualizing the proteins, there are some pretty basic things you should be aware of. One thing is that the protein models can be seen as kind of hierarchical systems, so that there are multiple chains, often different polypeptides, maybe a chain of protein and a chain of DNA, and the DNA actually would be 2 chains to make the double helix. Each of the chains is comprised of residues. We have amino acid residues in proteins and nucleotides in DNA and RNA, and then the residues are put together of atoms which are bonded to each other to create the molecular structures of amino acids and eventually the full chemical structures of proteins of thousands of atoms. Now, there's nothing mysterious in the PDB files. They don't give you anything that goes to inverse space or lists any of those diffraction intensities. The final result is basically just a number of points representing the atoms, and there's a regular XYZ coordinate position given for each atom. And then we have to know about atoms and het atoms. Not all atoms were created equal in the eyes of crystallographers and PDB managers. We'll see that in a couple of slides very soon. And we don't have all the atoms, so even if amino acids have a lot of hydrogen, hydrogen is mostly invisible in crystallography, so they are not present in the models either. They only represent the heavier atoms, that is nitrogen, oxygen, carbon. Only when we go to very good resolutions like 1.2 angstroms and below, so we start seeing hydrogens. Now, the atoms in PDB are, the term atom is designated for atoms in amino acids and nucleotides, so only the DNA and RNA and protein part is considered to be real atoms. And in the text format of a PDB file, we can see how two amino acids are represented as atoms in one PDB file. This is 1-BTK for Bruton's tyrosine kinase. And you can see the coordinates here. These headings are not present in the file. I've just added them for teaching purposes. We have the positions, the coordinates, in which the coordinate system is in an angstrom scale. Then we have the residue number in the chain. We have residues 4 and 5, which are valine and isoleucine. And we have the atoms in each, the atom type. We have the main chain atoms, amino nitrogen, alpha carbon, carbonyl carbon, carbonyl oxygen. We have the side chain atoms, beta carbon, and the 2 gamma carbons after the branching point. So you can almost build a mental picture of the amino acid by guessing what these atom names mean. And the same is true for isoleucine, except that the branching pattern is different. We have a branch of 2 carbons and a branch of 1 carbon on one side. All the atoms are numbered sequentially, so this is relative to this PDB file, and we have some technicalities like the B factor, which is a measure for the mobility of each atom, or rather the certainty in its position. Small values meaning that the electron density implies a better known position for that atom. Now the het atoms are all the non-protein, non-DNA or RNA residues. And whereas those standard components of protein and nucleic acids have just a small number of residues with standard names, so these heteroatom groups can be just anything. There might be a drug which is still not synthesized today, and next year somebody will be making a crystal with that drug bound to a protein it's targeting, and they have to figure out what they are going to call that new molecule in PDB in a limited space of 3 characters. And now, as an example, we have something going towards the end of the same PDB file. We have 4 zinc atoms, and then we have multiple HOH residues, and yes, you guessed it, this means water. Regarding chains in PDB files, I mentioned that there can be strands of nucleotides or they can be polypeptides. And if you have multiple chains, not just a single polypeptide in a PDB file, so you have to distinguish between the chains as A and B. You may have seen A and B in the previous slide. And the protein numbering usually restarts from 1, so with 2 copies of this 1 BTK or the BTK protein, we might have residue 5 in chain A and residue 5 in chain B. And why do we have several chains in one PDB file? The biological explanation might be that we have crystallized a real dimer of proteins or a larger complex, and so each part is then labeled as a separate chain. And sometimes in crystallization you get the crystallization happening so that we have a unit cell which is repeating itself over and over through the crystal in 3 dimensions. It could be a cubical unit cell or rhomboidal one or many other types. But the point is that this unit cell might not contain just a single copy of the single protein, but there might be kind of pseudodimeric arrangement within the unit cell. And if the 2 copies or 4 copies in the unit cell are not exactly identical, so you get a better model that more accurately represents the measured data if you have these multiple copies which are near symmetrical but not quite. And so these crystallographic copies might not be actually binding together, and you might be able to see the difference once you open several such copies, so that you see that if there is a lot of water between the protein subunits, so they are probably just together in the crystal, whereas if there is a very tight interface, so it's probably a real dimer. But anyway, PDB authors help you. They are trying to present the biological assembly, which shows if it's a monomer or dimer or what, which would be seen in solution or in biological environments. And then they give the asymmetrical unit, which shows the arrangement of subunits in the unit cell. But anyway, when you find a model of whatever you are looking for, one of the first things you should learn is how many chains does it have? Are the chains all the same protein, same polypeptide, or are they different? Is it a heteromer or a homomer of multiples? And in case of the same protein, are these actual biological multimers or just crystallographic things? Then the residue numbering might give you some surprises. So there might be gaps in numbering because some parts of the protein may be invisible. There might be a flexible loop which you can't find in the electron density. So it is in the sequence, but it's missing from the actual model. And then there might be gaps in the numbering if the authors think that they need to follow a canonical numbering, which is based on a model prototype protein. For instance, for immunoglobulins, they follow canonical numbering so that the constant parts of the Ig chain have constant numbers, whereas in the variable loops they have canonical numbering, and then depending on the length of the loop there might be gaps in the numbers, or there might be insertions so that a long loop is created by putting multiples of the same number, 85A, 85B. And then even if we have regular linear numbering, you have to find out if it's numbering which is directly as in UniProt, which is luckily the most common, or whether the authors decided to start one from the end of the mature protein in case there is the removal of signal peptide to create a shorter mature form. If you do a search in PDB and you have multiple hits and you want to evaluate which one you should be looking at, so first of all, you should first confirm if your keywords actually led you to the actual protein you wanted to have. And there's a lot of mutant or variant proteins in PDB. After the normal wild-type protein has been crystallized, there's no added value in producing more crystals of the same. So if you know how to crystallize something, and if there's any interest in variants, so why not make those engineered variants, do the crystal, and see if the structures are altered. If you can correlate the variant structure to increased or decreased enzymatic activity, so that makes interesting science. And so you need to spend some time in reading the PDB description, maybe looking at the article to find out if this is actually what you need. Of course, if you have controlled vocabularies like gene names, so that helps you in finding exactly the protein you want. But then the models in PDB might not be of entire proteins. It might be just a small domain in a longer protein, and you just have to figure out which part of the sequence is covered in the protein. And UniProt is good in that it's giving the positions in the UniProt sequence numbering for all the protein structures it knows about, all the protein structures from PDB which have been cross-linked to UniProt. Then regarding quality, we talked about resolution and R factors, so you might compare them to each other, and there are slider graphics which we'll see in the demo, which are indicators of various quality measures. And comparing all of these, you should be pretty soon confident which of the 5 alternatives is the best one. But if you have 100, so you should make some shortcuts, like just looking at the best resolution first, and once you find something that covers what you want, so take that, and that will probably serve for your purposes. Okay, in addition to finding out about the number of chains and identity of chains, whether they represent the same protein or different one, there might be some heteroatom groups, like what are those zinc atoms that were listed for BTK. You might find out about that looking at the article. Or if nothing else helps, so open the actual structure and try to see, are they bound in the core of the protein, or are they on the surface of the protein, or what? Yeah, and as a hint for finding out about what kind of numbering you have for your model, there's a sequence tab which allows you to call up the PDB numbering and UniProt numbering and compare if they are the same or different. Now, there are many strategies you may use for finding structures in PDB. One of them, the straightforward one, might be to do a keyword search in PDB. That is not that simple. I've been struggling with the keyword searches. I seem to be getting all kinds of irrelevant hits, maybe because I've been using too short words or abbreviations and not using the advanced search, so I get all kinds of false hits. The advanced search is not that nice in my opinion, so even if you get better results, it's not that easy to use, but anyway, it might be worth the effort if you really want to have the models and all the most recent models. And the advanced search starts to get better when you have to start sorting your results by resolution. You can decide to have just one range of resolution, let's say from 0.5 to 1.5 to have just the very best. You could start from UniProt. If you find your protein of interest in UniProt, you can look at the structure crosslinks if there are any. But the updates might not be totally on a daily level, so you might not see the most recent structures in that way. But then if you are looking at the UniProt entry and you see that, oh my, there's no known structure for this protein, it could well be that the autolog protein from mouse has been crystallized, even if the human protein is not there. And the mouse protein can be used for building a fairly reliable model of your human protein. So trying to find related proteins which are close to your protein of interest is a useful approach and a good question to ask. And you already know of the tool for that purpose. That would be blast searching. So just do a blast, but select the PDB protein sequences as your target database, so you'll get the answers. You can choose PDB as a database to search in NCBI, or you can go to the sequence search tools in PDB. You have to take search and then sequence search, and that will take you to a BLAST-like approach, which I think would be using multiple algorithms. But anyways, this is a recommended approach in that it finds you matches to all parts of the sequence you use as the query sequence, and if you are only interested in a particular part in the sequence, you can use the subsequence as your query and get some preliminary filtering of the results. And the only awkward situation is when you are looking for something that has a lot of models. For instance, CA2 has 886 crystallographic structures in PDB, human CA2, and trying to go through all of them is a lot of work, and BLAST really doesn't help you because you don't get the resolution information in the BLAST results, as far as I know. And then if you get those kind of off-target hits, you have no exact match, but you find something that is 70% identical in sequence. So that's nice because you can use it for homology-based model building. And we'll talk about that on Monday. We may have 5 minutes for that. Now, let's have a look at hands-on exercise in PDB, and we'll use a protein called P53, a tumor suppressor protein, as our search target. Here we are on PDB homepage, ready to start searching for P53. Now, if we just click on the magnifying glass without any limits, that would give us 5,000 entries, which is probably more than real P53s here. So we'd better go scroll down in the options here. We have many different fields where we can try to find P53, and P53 in gene names sounds like just the thing we want to find. And now this is a much better search. We have just 208 structures. And now we need to get the left hand side of the page to view. We want to do limits like going for human sequences, and maybe we want just X-ray diffraction and just the best 3 classes not going above 2.5 angstroms resolution. And also we'd like to skip the oldest ones which might not be of such great quality, so let's stick just everything from 2005 and onwards. And then clicking on the magnifying glass for refinements, we should get the filters applied, and that's 143 structures. And now let's choose something to have a model to look at more closely. So for instance, one of these which is listed as a mutant with multiple 1, 2, 3, 4, 5 different mutations in the sequence. Now on the page of the PDB entry we selected, we'll find the slider graphs we discussed, which show different measures of quality as percentile ranks, meaning the relative position compared to other entries in PDB. On the right, in the blue side, it's better, and on the red side, it's worse. And the closed bars are percentiles relative to everything in PDB or relative to X-ray structures, and the white bars are relative to X-ray structures of similar resolution. This is 1.8 angstroms. So you can see that this is quite good in some respects and pretty average in others relative to everything in PDB, but then relative to other structures of the same resolution, this is actually doing a little bit worse. And if we scroll down to sequence information, so we can see that this is 200 something residues in length, and now the reference sequence is set to be the PDB entry itself, but we can choose it to be the UniProt entry. And now we can see that this is coming from the middle of all of P53 sequence. But this still doesn't tell us how the entry is numbered, whether we have PDB numbers or UniProt numbers. So we need to look at the entry itself, which we can do by downloading files. And if we go for the PDB format file, we can download it and save it into our downloads folder and open in a text editor. This is PDB format in the text form, not in the XML or JSON format, and this is pretty nicely human readable, and we can just scroll down to see what we find of the sequence. And we can see that there are some missing residues, so that residues 94 and 95, and some in the end are missing from chain A, and likewise for chain B. So we have 96 to 260. This is UniProt numbering. And now if we look at the actual atom lines, there we go. So you can see that the first residue for which we have atomic positions is serine 96. We also discussed the idea of as asymmetric units and biological assemblies. And now the view PDB is showing to us seems to indicate a monomeric biological assembly. And there's another biological assembly as a monomer, which is probably just a second copy in the asymmetric unit. And let's see if there are more. No, it is just two. And we can see that the unit cell contains these two copies of this fragment of the p53 protein, which would be the p53 core domain, as they call it. And from the distance between these monomers, I can tell you that this can't be a real dimer. There is not enough protein-protein contact to make this a dimeric thing. So the space in between is mainly filled with crystallographic water or crystal-containing water. It is typical for protein crystals to be 50% of water and 50% protein. So these are just dimeric or two chains in the unit cell for crystal packing, which is slightly asymmetrical to give us two different copies in the unit cell. If in different conditions we might get a crystal where we could reduce the unit cell into just one asymmetric unit, but don't take me too seriously. I'm not a crystallographer. I've read one book on crystallography, and that was long ago. Next, we will be comparing the data we found in PDB to the structural data we find via UniProt. This is the UniProt page for human p53, and I'll click on the structure sidebar link here. And we can see that there are really lots of different models here, and we have some which are located in the very end of the model. These are unfortunately alphabetically by the PDB entries, and there's no way to change the sorting order here. So there are many which are representing the core domain, and then quite a few that are for the N-terminal 300 plus sequences. But the core domain is the very largest one, and now for some of the N-terminal fragments, these are really short, just like 16, 17 residues. And the one we had chosen to work with was (2J1Z). And we can see that there are quite a few entries that are coming with 2J something, probably coming from a same article. And now we have lots of these core domains here with nearly consecutive codes. And this 2J1Z is the one we wanted. And let's see if we can find the total number of structures reported for for this P53. Come on. And unfortunately we don't get the numerical data, so we have some good things here that we can find all the sequences, compare the resolutions and positions easily, but whether this is everything we found in PDB is not directly available at a glance. What we learned in the previous demo was that my memory of using the sequence tab for comparing the UniProt numbering and PDB numbering didn't quite work out, but instead we had to look at the sequence as it was shown on the structure summary page, and we could see the UniProt numbering by taking the UniProt entry as a reference there and then looking inside the entry and comparing it to the UniProt entry was one way of confirming that we actually had the same numbering. But this could be done even inside of Chimera. But let's move on to the last topic, which is different ways to represent molecules. There are many different graphical styles, and some of them are better for highlighting the overall shape of the molecule, whereas others focus on placements of atoms and connectivity. And in case of proteins, we can just do a symbolic tracing of the polypeptide to just show what is the run of the polypeptide backbone and what are the secondary structures. And when we do actual finished images of proteins, we might want to use combined presentations where most of the protein is in, let's say, ribbon representation so that there is not too much detail shown, and show the atoms and bonds only for maybe the active site, so that when you see clear atoms and bonds, you know that there's something special going on there. Now, the elements in chemistry have their typical colors, which are shown here. There's red for oxygen and blue for nitrogen, which correspond to the basic and acidic colors of your indicator paper. Sulfur is usually in yellow because sulfur as the element powder is yellow. And likewise, phosphorus is kind of yellow or orange in its physical appearance, and that's the same color in the molecular graphics. And then carbons in the upper image are shown in gray, and they are darker gray or black in the lower image. But if we have some really strange off color like green or cyan, that could also be an indication of carbons. And hydrogens, when present, which they are not in most PDB structures, they would be in white. And for metal ions, we don't have any rules, so if there's a zinc or magnesium, it can be just anything, and when you make your own visualization, you don't break any rules if you choose any color for something like such. Now, the atoms and bonds, as we see here in the bottom, are pretty clear, and if we have this kind of a stick representation, you have to understand that in every corner and every end of a line there is an implied atom. And in this example we have the atom labels. It's a terrible visualization. It's so hard to get good stuff nowadays, so it shouldn't be really here. But anyway, this is good because we can kind of see past the atoms. There is no hole inside a 5-membered ring, but it would be totally filled, as we see in this example. this ring is really with atoms touching each other, not with a hole. But if we have like a full protein, it's not a good idea to show all atoms and all bonds. It gets kind of messy. Now, another possibility to avoid the problem of missing the corners as atoms is balls and sticks, where we have tiny balls smaller than the normal idea of atomic radius, but still something to symbolize that there's something there. And now if we change into a space-filling sphere representation, this is exactly the same tryptophan side chain in space filling and in balls and sticks. So you can see that this is definitely hiding a lot of stuff behind it, and if we just want to use a symbolic view, so this would be a better version. But if we want to show how much space this particular side chain takes, so this is much better. But anyway, it's clear that we can't really see very well the bonding pattern, that we have a 6-membered ring and a 5-membered ring here, which are joined by 2 atoms to connect to the peptide backbone. Now, the space-filling spheres are okay for indicating the space taken, so they work even for a larger protein. This is about 200 amino acids. But these are kind of awkward in that a molecular surface is not certainly consisting of spheres, and certainly not of nice bright colored spheres. So if we want to simulate a water accessible surface, we could do a smoothing of these spherical displays and then show it as a surface. And here's a comparison of the previous sphere diagram and then a smoothed surface. And in addition to bringing a more realistic shape, the kind of 3-dimensional perception is better, so we can see more clearly where we have cavities in the model, which is not that clear in, it's not as clear here in the sphere diagram. But what we miss is that we don't see the oxygens and nitrogens in the sphere. In the surface. Then the ribbon presentation or cartoon representation for the polypeptide. This is the textbook way of showing proteins, so that you see how the proteins are folded, how the polypeptide runs through space, and where are the alpha helices shown as spirals and beta strands shown as arrows. The arrowhead shows the direction from amino to carboxyl, and so the whole polypeptide is easier to follow, and we can see that this would be the last residue of this whole protein. And there seems to be a break somewhere, because we have several other ends here as well. Or maybe this is a dimeric protein. Well, anyway, the meaning of this illustration is just to show you secondary structures and the ribbon representation. And now if we have surfaces, we might combine these things to put a solid ribbon inside a semi-transparent surface. But this is a teaser image. We learn more about transparency when we start using Chimera and make our own visualizations.